In this tutorial, we will look at digestion of carbohydrates. Now, in our daily diet, about 40 to 45% of the daily caloric intake is from carbohydrates, and about half of the carbohydrate calories in our diet is obtained from plant starches. So let's look at some of the basic molecules that are present in carbohydrates. In a typical diet, we have monosaccharides. Monosaccharides are simple sugars that cannot be further broken down by hydrolysis. And the examples of monosaccharides are glucose and fructose. Here is a molecule of glucose, and here we see a molecule structure for fructose. Note that we get glucose from fruit or honey or grapes. Similarly, we get fructose from fruit juices and honey. Next, we have disaccharides. Disaccharides, of course, contain two monosaccharides, and they can be broken down into those two monosaccharides. Examples are lactose, as the molecular structure shown here, and sucrose. Again, the structure is shown here. We get lactose from milk and milk products, so from dairy items, and sucrose is the plain table sugar that we get from sugar cane or sugar beets. Next, we have polysaccharides. Now, polysaccharides are long chains of polymers composed of monosaccharide units that are bound together by glycosidic linkages. We will look at these linkages in the following slide. But the examples of polysaccharides include starch and glycogen. Let's look at starch. There are two types of starch molecules. One is called amylose. Amylose is a straight chain glucose polymer. This is where the glucose molecules are connected via the alpha 1,4-glycosidic linkage. And here we can see a structure of two glucose molecules connected together with the alpha 1,4-glycosidic linkage. And of course, similar linkages extend on both sides, so it is a long chain. Then there is the amylopectin. Amylopectin is a branched glucose polymer. So in this case, we have branched molecules that are connected via alpha 1,6-glycosidic linkage. And again, the molecular structure for amylopectin is shown here, and we see how these branches form the alpha 1,6-glycosidic linkage. Now, amylopectin is a much larger molecule. It comprises of about 10 raised to power 6 or a million glucose units per molecule. The average chain length in case of amylopectin is about 20 to 25 units. About 20% of the starch in our diet is made up of amylose, and the remaining 80% is in the form of amylopectin. Some of the common sources of amylopectin and amylose include corn, potatoes, rice, and bread. Now, starch is a plant storage polysaccharide in contrast to glycogen. So glycogen is found in animal cells, and it is a polysaccharide storage molecule. Its structure is similar to amylopectin, but it has a lot more branch points. So we have seen some of the basic molecules in carbohydrates for monosaccharides, disaccharides, and polysaccharides, and we have also considered the two types of starch, the amylose and amylopectin, and glycogen. In a following tutorial, we will look at how these structures play a role in the digestion process.